Can you read a cell's mind? Science isn't just mixing regions and solutions, but sometimes requires the operating of heavy machinery, which seems straight out of a sci-fi movie. These machines can analyze our cells and proteins. But that made me think, how can we identify our cells without directly looking at them? Enter Dharshini Raju, today's episode spotlight. Dharshini works at the flow core, and today I shall see her command the flow cytometer and help me understand the answer to this myth. You can tell everything about a cell just by looking at it. Dharshini starts her day early, setting up these incredible machines. But first, coffee. Black, of course. She checks the day's schedule and experimental goals on Connect, then powers up the cytometers and making sure the lasers and fluidics are ready to run. From 10 o'clock, the real work begins. Sorting and analyzing cells for scientists across the center. Dharshini watches scatter plots and fluorescent signals in real time, ready to troubleshoot the moment something looks off. Between runs, she trains new users and supports researchers with data analysis, using software like FlowJo to gate populations, compare controls, and turn dots on a screen into meaningful insights. The day often ends with project meetings, cleaning, and shutting down the instruments. By 6, the lab is quiet again, until tomorrow's experiments. But what do these machines do? And how? Upon close observation, I think I cracked the code to flow cytometry. Imagine you have a smoothie made of different fruits. Bananas, strawberries, blueberries, and you want to know what's in it and how much of each fruit is there. Flow cytometry is like a super smart juicer that can take each tiny piece of fruit, shine a light on it, and tell you what kind of fruit it is, how big it is, and what's inside of it. She supports the researchers with their projects, advancing the great age of neuroscience through many techniques. One of them is the staining protocol. Upon my observations, I found out that a successful experiment on a flow cytometer starts long before the machine is switched on.
It all comes down to how well the samples are prepared.
Here in this core facility, these techniques are used to study some of the brain's toughest questions, from how immune cells respond in Alzheimer's and Parkinson's diseases, to how brain cells react to new drugs or have changes in their synapses. Every dot of color on the screen tells a story about what's happening deep inside the brain. Flow cytometry isn't just sci-fi cell counting. It's so much more. You can tell everything about a cell just by looking at it. But when we decode it with colors, it's the closest we've come to reading a cell's mind. Behind the lab bench, there are supporting roles in the form of experts like Dharshini, who play pivotal roles, showing that cells don't just speak through shape. They shine with data that can be crucial for us to understand our brain. Because behind every dot on the graph is a story waiting to be told. This is the wild lab life. And this is just one of the many stories here that are a part of the great age of neuroscience.